Next, I'd like to invite to the stage Dr. Jansson, Senior Staff Scientist at Lawrence Berkeley National Laboratory's Earth Sciences Division and Adjunct Professor in the Department of Plant and Microbial Biology at University of California, Berkeley. Her research focuses on understanding key functions carried out by microorganisms in complex microbial communities, such as those residing in soil, sediment, and the human gut. One specific question she is trying to understand is how a warming climate will impact microbial decomposition of carbon stored in permafrost. Janet is a former Vice Dean, Faculty of Natural Resources and Agricultural Sciences at SLU, Sweden, and a member of the Swedish Royal Academy of Sciences. To discuss the issue of thawing of microbes in Arctic permafrost, please help me welcome Dr. Janet Jansson.
Thank you very much for that introduction, and it's really my pleasure to be with you today and have an opportunity to discuss some of our research that we're doing in my laboratory. So we will be moving in my talk from the oceans to the soil. And this is an image that was taken, actually I took this image in Greenland, and it is showing some of the permafrost areas in Greenland. So much of the Arctic, the northern hemisphere, is underlain by permafrost. So permafrost can be defined as soil that's been frozen for at least 2 consecutive years. And the purple regions that you can see here on this map are the areas that have permafrost underlying them. And you can see that the northern hemisphere is really dominated by permafrost regions. The darker purple land surfaces are underlain by continuous permafrost, and the lighter purple is discontinuous permafrost. I've also indicated some of our sampling sites, and I've been very fortunate to be able to get samples from Alaska, a couple of different areas, also Greenland, Svalbard, Norway, and 2 sites in Greenland, actually. So this is the situation. This is where the permafrost is. If we also look at another map of the globe, this is colored differently. So the darker colors here show where the most carbon is currently stored in the soil. And you can see that this coincides quite nicely with the previous map that I showed you. So the darker colors show that where the most carbon is, and that's really where the permafrost is. So what this means is that most of the terrestrial carbon on Earth is stored in permafrost, frozen permafrost. So what are the implications of this? The climate is changing, and we can see evidence of this in the Arctic. The Arctic is a very vulnerable landscape, and as it's warming, there are many different kinds of landscape features that occur. So some examples are shown in this diagram here. We have an increase in the temperature, of course. That's causing melting of glaciers. It's also causing coastal erosion. Shrinking lakes. These thermokarst features are when the land subsides and forms a lake, and it can sometimes be a bog as well. These kind of strange formed landscape features are called polygons. It's polygonal ground, and that occurs because of ice wedges that form between cracks in the soil, and they get deeper and deeper and then make these polygonal features. There is also thermal and fluvial erosion, and if we look at a cross-section of the landscape, this is the area, this is my view of the landscape. So you have the permafrost, which is this lighter color, and it's overlain by an active layer that is seasonally thawed and frozen every year. So that's called the active layer. And the permafrost is continuously frozen. So what are the microorganisms doing there? I'm a microbiologist, so I'm interested in the microorganisms. Well, this is a model that from NSF that is predicting what is going to occur with climate change. This is one model that I particularly like. It shows that if this is the current situation where the carbon balance is neutral, so what comes in from the atmosphere is equalized by what is respired. So you can see that there are ice wedges in the frozen permafrost. Then there are plants growing in the active layer. they respire, they release carbon dioxide into the atmosphere, but also some is taken up by the plants. So this would be a net zero effect. In the short term, as the climate gets warmer, it's predicted that the plants will also grow better. That means that they can take up more CO2. So maybe in a short term, like 15 years, due to the plant growth, there will be a net intake of carbon dioxide from the atmosphere into the soil. The plants are still respiring carbon, and the microorganisms in the soil are also respiring and releasing carbon dioxide. But it could be more in than out. However, what's really scary is the long-term impact. And this is not that far away. It's 35 years plus. When the active layer sinks and the permafrost thaws, so that old carbon is accessible to microorganisms, and they start to degrade this old stored carbon and release that into the atmosphere, then the net impact will be an efflux of carbon into the atmosphere. So here is the microbial, my view, again, the microbial view. So we have the active layer, the permafrost, and the types of reactions that are carried out by microorganisms. So there is organic matter in the soil. I told you that already. It's stored there. And it can be fermented and respired by microorganisms. And they can produce either CO2, which is the greenhouse gas that we're very familiar with, or methane. Some microorganisms produce methane. Now, methane is 20 times more potent as a greenhouse gas than CO2. And then there are other microorganisms that can uptake methane or CO2. So it's the net flux of these gas exchanges that's really important. So as the permafrost thaws, the microorganisms are gaining access to this carbon that's been stored in the permafrost, thereby releasing greenhouse gases, methane and CO2. So the dilemma, and this is still the dilemma that we have now, that this is Leonardo da Vinci's quote from the 1500s, and we could say the same thing now, that we really know more about the movement of the celestial bodies than about the soil underfoot. So when you look at soil and try to understand microorganisms, it's really, here I call it Earth's dark matter. It really is Earth's dark matter. It's a big unknown world in soil. Just a single teaspoon of soil, a gram of soil, contains billions of microorganisms, but the vast majority of those have never been cultivated on media. We have no idea what they're doing. So over 90% are unknown. And these microbes are responsible for the cycling of carbon and other nutrients for our whole biosphere, for the Earth. And it's not known how climate change will impact microorganisms that are currently living or residing in permafrost. So what our group and other groups are currently doing is trying to understand microorganisms in soil, such as permafrost, by bypassing the need to cultivate them. And this is what I call an omics pipeline. It's a series of molecular methods where you can directly extract DNA or RNA or proteins or metabolites just directly from the soil and then sequence the DNA and RNA or run it on a mass spec to see what the proteins and metabolites are. And it doesn't matter if the organisms can grow or not. So this is our approach, is to determine the hidden diversity of microbial communities at different levels. And each of these steps in this omics pipeline gives you different kinds of information. So if you sequence DNA, you see which genes are there, and that tells you which microorganisms are there and what genes they have. If you sequence RNA, you will know which genes are expressed. If you look at the proteins, you can see which proteins are produced. And if you look at the metabolites, that gives you more information about the biochemical pathways that are being used by those organisms. So our research aims are to determine the impact of thaw on permafrost soil communities, the microbes, and to understand their carbon cycling dynamics and how those are impacted by climate change. And ultimately, this information should improve climate models that are deficient, that they don't have enough information about these processes. So a few tidbits of data. This is real recent data. There's microbial life in permafrost. This is very recent data. So even at sub-zero temperatures, some microorganisms are alive. And using these molecular tools, we can identify who they are. We can see, this is looking at phylum level taxonomic descriptions of the organisms that we find. So each of these colors in these bar charts corresponds to a different microbial phylum. And we can, when we look at the DNA, we can see that there are many different types of microorganisms in permafrost. That's the DNA. When we look at the RNA, we can see some are more actively expressing genes than you would predict from looking at the RNA alone, such as this orange phylum here that corresponds to firmicutes. They're more active than you would predict if you just looked at the DNA. They're expressing a lot of RNA. And some are less active. They might be dormant. They might be just sitting there growing very slowly, or dormant, and they don't produce very much RNA. Also, when we look at the proteins, we find a lot of proteins in permafrost. And some of those proteins I've mapped onto this cell map are for iron reduction, which gives us a hint that iron reduction might be a process that the organisms are using to be able to survive in sub-zero temperatures in this particular sample that we're looking at. And we are also isolating microorganisms, because even though we're using all of these molecular tools, it's also interesting to try to isolate into culture and understand the microorganisms that we can grow on media. And this is an example. I think you can see this long, stringy thing here. That's a novel methane-producing archaean that we've been able to isolate, and then we sequence that and can understand its properties. This is another example of recent findings where we did a short-term thaw. We incubated permafrost that was frozen for 2 days at 5 degrees and then saw what happened. And there was, well, first of all, this is just mapping the genes that we see when we sequence all the DNA onto biochemical pathways. This is just a small portion of a pathway map. And all of the pathways that are in red are enriched in the seasonally thawed active layer that freezes and thaws every season, and all of the ones that are in green are enriched in the permafrost. So you can do a direct comparison and see that these two environments are different. The permafrost pathways are different. Now, when you do a short-term thaw, just after 2 days at 5 degrees, there is a very rapid shift. This yellow, these two yellow shaded areas here represent permafrost, 2 different permafrost samples. They were taken as cores that were incubated for 2 days, and the genes that are representative of that community shift very rapidly. This is the 2-day composition of the genes. So, and they're also quite different from each other at the beginning. These 2 permafrost samples were very different. They were frozen for thousands of years and they diverged. But after a short-term thaw, they became very similar to each other. And the day 7 samples are even more similar. And these that are circled here are the active layer samples. They don't change a lot when you incubate them at 5 degrees because they're subjected to that every year anyhow. That's our hypothesis. They freeze and thaw every year. So what we think is happening is that a short-term thaw causes a rapid shift in permafrost microbes. Now, this is a little scary too, because this was not the dogma that was thought to occur. Until we published these results, it was thought that if there was a thaw, it would be a very gradual shift in the microbial communities, but it shifts very rapidly. And we think if we had kept doing this a longer incubation, we would have shifted the entire communities to be more similar to the active layer. And also from this data, we also were able to get a genome for a novel methanogen, a methane-producing archaean, which was a nice little bonus. So that's the data that I was going to show. I just wanted to mention in summary a couple of ongoing projects that I'm currently involved with, and there are other examples as well. But it's very gratifying to me to see that this is very seriously taken by the funding agencies. These are very large, multi-team, multidisciplinary endeavors that are focusing, in this case, in Barrow, Alaska. This is the aerial view of those polygon landscape features that I was mentioning to you, where the whole team is going in. It's funded by the DOE, going in there and looking at the microbiology, but also the hydrology, the geochemistry, and trying to understand what's going on with these thaw features, and then to use that information for models. The other example is the Danish Center for Permafrost Research, CENPERM. That's me there on a boat. I'm the only girl. but that's an iceberg in the background. But that's looking also at permafrost degradation on Greenland. And so if anybody's interested in either of these projects or anything else, I'd be happy to discuss at the reception. Thank you.